All right, so it is my pleasure to introduce our next speaker, Dr. Gabriel Golczer-Gatti. So Dr. Golzcer-Gatti was born in Caracas, Venezuela, and spent most of his childhood years on a Caribbean island off the coast of Venezuela called Margarita Island. His passion for genetics brought him to obtaining his biology degree at the Universidad Simon Bolivar in Caracas, where he subsequently graduated with honors from the Master's in Biological Science program in 2012. He continued his education at Tufts University in Massachusetts, where he got his Ph.D. in 2018. Up to that point, his focus was to apply genetic tools on non-model insects to various questions in the field of taxonomy, population genetics, ecology, and evolutionary biology. He did both the experimental work at the bench and computational analysis in R, Python, and used several other bioinformatics tools. At the end of his PhD, Gabriel decided to pursue a career only in data science, focusing on genomics, epigenomics, and transcriptomics. He continued his training doing a postdoctoral fellowship at Harvard Medical School and the Massachusetts General Hospital Cancer Center, working with renowned scientists both in bioinformatics and the cancer fields. After a year and a half, he transitioned to the biotech industry, working at CAMP4 Therapeutics as a data scientist, continuing his journey of uncovering mysteries in biological data in the field of gene regulation. So it is my pleasure to welcome Dr. Golczer. Thank you for coming.
Thank you, Laura, for that introduction. So I'm gonna go right away and jump in. So the title of my talk is From the Caribbean to Massachusetts and How NR4A1 Regulates Expression and Suppresses Replication Stress in Cancer. I'm going to use the Mass General Cancer Center and Harvard Medical School template because this project was my main project as a postdoc, although I no longer collaborate with them. So Laura already mentioned where I'm from. I'm from Venezuela, just north of Dr. Papas, who just spoke. Specifically, I grew up in a Caribbean island, and this was kind of the setting that I spent my weekends. So you can imagine that growing up here will get you drawn to the ocean and be a surfer of some kind. But when I was in high school, I didn't want to be that, not even a marine biologist. I wanted to be a geneticist. And that drove me to study biology because there was no like genetics degree per se in Venezuela. And when I started going through all the biology courses, I was more passionate about analyzing lots of genetic data over everything else, over like doing PCRs or spending time in the lab. So I was so passionate that I joined the only lab at the time there that worked with tons of genetic data. And that lab was working with this insect right here to fly. This ruler scale is a millimeter scale, so it's like roughly 5-6 millimeters long.
Dr. Golczer, we can actually see your slides.
Oh, sorry.
No worries.
I forgot to do this. Okay. So yes, so these guys, can you see the laser pointer now? Yes.
Yes.
Yep. Okay. So this guy here, pretty small sandfly, and yeah, I had to go to the jungle, tropical jungle, to capture them. This is me under this light trap here. And the interest of these insects was that they transmit a disease parasite called Leishmania right here in this microscopic picture. So all this work in the field and the lab to extract DNA to get this. And this is Sanger sequencing, not used at least on a broader scale because we have now next generation sequencing, but I spent tons of time analyzing this data. And this kept me passionate and going into bioinformatics. So for the next steps, I went to do my PhD here in Boston and joined a lab that was kind of working with insects as well, with genetics. And coming from this climate to this climate was quite a transition. And this is the insect that I work in during my PhD. So it's the European cork borer. You can see the caterpillar here in the stalk of the corn, and the adult form here. So also extracted DNA, doing analysis, and uncovering some interesting evolutionary biology and population genetics questions. This journey more on the Q&A section, so I will go to the title of my talk, which focuses more on NR4A1 and cancer. So before I start with this story, I'm just going to go and state a disclaimer that I'm not the typical bioinformatician that you might find in some labs in the industrial or academic setting that just like, oh yeah, give me the data and I churn it out. And I don't talk to the people at the lab. I'd like to collaborate a lot with the lab and bench people and go back and forth with them, design questions, like try to get the experimental design right. So when it makes sense, when the data comes in, I have a better idea of how to analyze, and we don't have to go back and forth afterwards and then say, no, we cannot repeat the experiment. We already did it. We spent $20,000 on it. So I prefer to be in the early stages on it. And this project, I had the pleasure to collaborate with Hong-Shan Guo, Ph.D., who was the postdoc fellow at the time in this project. So another disclaimer here, this project has been recently published last year in Molecular Cell, so you can find it there. And you might find the same figures or not find the figures that I'm showing you here because they were intermediate and they work better on a slide than on a tiny space on the figure panel on the paper. So jumping right in, I should have started with cancer. So 1 in 8 women in the US will develop breast cancer specifically in her lifetime. And if it's caught early, as you can see in this bar plot, you have a pretty good chance to survive and overcome it. So the problem is that when it gets stage 3 to stage 4, it starts to do metastasis and the cancer cells can go to other tissues or organs, organs like the brain, lungs, ovaries, major organs. So we want to understand how the metastatic process works. And this gets us to circulating tumor cells. So in the early stages, when you have a primary tumor, then this tumor can shed into the bloodstream cancer cells. And these are called circulating tumor cells, or CTCs, as I'm going to call them from now on. And they can land in another tissue or organ and evolve into a secondary tumor, therefore creating what we call metastasis. So why are CTCs worth studying? Well, although you have 1 CTC per billion of normal blood cells, patients with CTC positive samples in the blood have worse prognosis. And CTCs may provide a real-time, non-invasive approach for tumor characterization and personalized therapy. So studying these CTCs can further understanding of the metastatic process and identify key drug targets to prevent it. The lab that I collaborated with, the Harvard Maheshwaran Lab at NIH HMS, has CTC derived from patients cultured in vitro. And with a multi-omics approach to understand the transcriptomic and epigenomic programs that drive these CTCs to produce secondary tumors, they started this project in 2017, 1 year prior that I joined as a bioinformatician. So this is the initial experiment model. Dr. Guo injected injections, these CTCs that are derived from patients, from breast cancer human patients, into these mice. And of course, we have imaging with bioluminescence markers to see where they are, what other organs or tissues they land in, and they form metastasis. And they cell-sorted the cells, separating mice from humans, and in throughput sequencing, single-cell RNA-seq, ATAC-seq, ChIP-seq. This is kind of a panel image of the time progression of the mice. And they injected 2 CTC cell lines, BRX-142, so from patient 142, and then BRX-82 from another patient. And you can see that starting when you inject into the heart, it goes all over the place, but it starts to concentrate in some major organs, in this case, the ovaries, and then later also in the brain, and also at some stages of the experiment, the lungs. So we dive right in the bioinformatics of it, transcriptomics. So if you see on the right the U map here of the transcriptomics, you see that I draw 3 clusters. And this first cluster has like the pre-injection, so cells before being injected into the bloodstream of the mice, and initial samples just after the injection cluster here. And then in later stages, from the first 2 weeks, they cluster here. So and then afterwards, so transcriptomic programs change over time. And when we compare in volcano plots the pre-injection per we found this little guy here, this gene, NR4A1, which is in the title of the talk. So I will try to explain the logic of why we focus on this guy. First, you see it's highly enriched when comparing day 30 or more samples and pre-injection samples. We also looked at the chromatin accessibility using ATAC sequencing. And here you have a correlation plot of the samples. We looked at overall samples, how do they look in similar or different to each other. And you might notice that the higher correlation is among the pre-injection samples, which are highly identical here and here, depending on the cell line. And there's a huge difference between those groups and the other groups, regardless of the tissue or the day after injection. So there's a change in transcriptomics and epigenomics here. Looking at the ATAC-seq more in depth, you can see in this heat map to the left, each column represents a gene, and the color is the relative accessibility comparing pre-injection and after injection, so after day 2. And you see that the genome becomes more accessible in these genes here at the bottom of these heat maps compared to the pre-injection. And we have NR4A1 here and another guy that I like to call the usual suspects in this paper, FOSB, EGR1, PTG2, June. You can also see them here in this plot to the right, which is a scatter plot when we want to compare both cell lines and the pre-injection and day 30 and above accessibility. And here you have no change in the center of these plots, and when you go to the corners, you have more open chromatin, or see these genes again popping up. NR4A1, FOSB, FOS. What are these? These are called immediate early genes in cancer. These genes are activating transiently in response to a stimulus or stimuli, and they are standing response mechanisms activated at transcription levels before protein translation. These are around 40 genes that include NR4A1, FOS, FOSB, PTG2, MYC, June, among others. We annotated the regions that gain chromatin accessibility. And we also did chip sequencing of using an NR4A1 antibody, comparing pre-injection and then the post-injection ovary mets, in this case, and brain metastatic cells in the case of BRX82. You might notice here this highlighted tracks, section of the tracks here. You notice that, for instance, NR4A1 gains accessibility, so the ATAC-seq signal is increased on the gene, not before the promoter or the promoter proximal region, on the gene body region. It happens also in FOS and in FOSB. And the ChIP-seq also gives us NR4A1 binding to the same region, roughly at the same time points. And NR4A1 ChIP-seq on both cell lines gave us a binding at mostly gene body regions, which is kind of slightly odd if you look at the literature for most of transcription factors. Here I have 2 heat maps on the left of each cell line, and you notice that NR4A1 is at the top because that gain accessibility, it's one of the top genes that have increased gene expression after the injection. And after doing including ATAC-seq, post and pre-injection ratio, and also transcriptomics, we found the top 6 genes are these genes here, and the one with the greatest scores is NR4A1. So we decided to focus on what NR4A1 doing in the metastatic process with this CTC. What do we know about this gene? It's also known as NR7, orphan nuclear receptors of family group A member 1. It's described to have putative ligands. These orphan receptors members remain to be identified, so not much information about it. The gene ontology often cites DNA binding transcription activity and sequence specific to DNA binding. And the latest paper at the time was Liu 2019, describing it as a key regulator in T cell dysfunction. It has paralogs, NR4A2 and NR4A3. So the first question after focusing on NR4A1 was, is this exclusive to CTC? So we repeated the experiment with another cell line, which is not CTC, it's also a cancer cell line. It's a triple negative cell line called NDA-MBT231, which I'll call M231 for short. And basically did the same thing, done a lot of sequencing, and then plotted pre-injection versus liver. We see NR4A1 popping up again, gaining chromatin accessibility, exactly at the gene body. Most of the gene body targets, majority of gene body targets. So it's kind of repeatable. We also look at the gene expression, and there's all. This is not exclusive to CTC. One is to have an in vitro model to further explore this mechanism, and we compare different kind of stimuli. We try X-ray, UV, heat shock, and measure the expression of some of the M231 cells and also MCF10A cells, and we found that the serum extravasation model mimic what we were seeing on the CTCs and the M231s injected into the mice. The model is basically you have cells growing in serum baseline, then you start them for 24 hours, you remove the serum, and then you replenish the serum and take samples 30 minutes, 1 hour, 3 hours, and 6 hours after replenishment. Then you do a lot of sequencing. So looking at the data here, Taxic, again, the plots comparing basal to 1 hour after replenishment for both cell lines that we tried that were not CTCs, NR4A1 gain accessibility. So we have the same pattern here. And the only common genes that gain accessibility between the two cell lines were NR4A1 and NR4A3. So we stick to NR4A1, we look at the expression, and you see in this bar plot that the expression keeps ramping up and then going down. So NR4A1 also binds to gene body regions when we do ChIP-seq. Most of the regions that it binds, 77%, it's gene body, and there's no promoter regions, which is kind of odd. So comparing our NR4A1 ChIP-seq done from MCF10A cells here, looking at the gene FOS and gene D, and comparing E2F1, which is a transcription factor well characterized. D2F1 is binding at the promoter or just before the promoter regions, but NR4A1 binds to the gene body and downstream of it. We also look at the gene ontology and GSCA and found that one of the pathways that were enriched is transcription for RNA polymerase promoters. So that prompted us to look at the RNA-pol2 chip data from these samples. And we see an overlap on it. We just see sometimes the peak of the typical peak at the beginning of the promoter region of RNA-pol2, but you see that there's overlapping between the NR4A1 and the RNA-pol2 in the same regions on these two genes and in others. So looking at the seed maps, we found that NR4A1 binding precedes the high expression. We can divide the genes into group 1 and group 2, the ones that have binding at the beginning of the serum, the baseline experiment, and then at 1 hour. So those will be group 2. And just before we start the cells, we have NR4A1 bound to this group of genes, which has FOS, gene B, gene, immediate early genes. And after the serum starvation ends, we start seeing expression increase in these genes. So we look at these three aspects on group 1 and group 2 genes. We have NR4A1 protein binding here on red. Then green, we have gene expression, and on blue, RNA-pol2. And when we have high protein binding of NR4A1, we have RNA-APOL2 traveling ratio, which is a measure of how RNA-APOL2 is transcribing normally or it's paused, is really low, which means the RNA-APOL2 is paused and transcription is low. Once NR4A1 is removed by the stress of starvation and then replenishment, transcription and RNA-APOL2 go up. So it's kind of, NR4A1 is kind of, for these genes, is stopping RNA-APOL2 for transcribing. And we can see that when we compare the genome tracks here. So this is kind of our model so far. NR4A1 is kind of blocking RNA-APOL2 when we have high coverage of the gene body. And then when we remove NR4A1, low coverage of the gene body, RNA-APOL2 transcribes normally. And we have more transcripts. We measure that with RNA-seq. And so this is kind of like putting a plug on a sink and filling it with water. The plug is NR4A1. You remove the plug, you have a lot of transcript, a lot of water pouring down the pipe. This effect with RNA-APOL2 is also found on BRX-142 and BRX-82 on our in vivo experiments. So everything lines up so far. So wanted to do, Hongxiang managed to CRISPR so creating NR4A1 knockout cells. And we measure, again, ataxy, chromatin accessibility. Almost no genes being outliers on chromatin accessibility, almost no change. Expression on the knockouts of NR4A1 of the immediate early genes is through the roof. It's the equivalent of when you add the serum back in the mechanism, the stress is no more. We have a high expression of NR4A1 on our cells. And this is how the knockout cells on this panel look like using immunofluorescence. And this picture gave us a picture, it's redundant to say, of what's happening. You see all these multiple nuclei. So the loss of NR4A1, the knockout, induces all genome stabilization. So the cells go haywire. So what happened to the mice if we inject NR4A1 knockout cells in vivo? Well, we have the N231 knockout control cells. We have this size of the tumors. Of the knockout cells, the ones that are CRISPR out, the tumors are smaller. So the tumor mass is significantly smaller once we don't have NR4A1 in play. And this also holds on with CTCs. Hongxiang couldn't do CRISPR on the CTCs, but he managed to do shRNA NR4A constructs, so knockdowns, not knockouts. And we again see smaller tumors on the knockdowns compared to the control cells in the same organism, in the same mice. So if we do the opposite, the overexpression of NR4A1, it favors cell proliferation and tumorigenesis. So a construct to overexpress NR4A1 gives us bigger tumors. So what is the mechanism behind NR4A1 and RNA-APOL2 pausing in breast cancer metastasis? We found out using S9.6 strip qPCR that there's formation of R loops that kind of stops RNA-APOL2. So TRIP-seq utilized a sequence-independent but structure-specific antibody to count massive parallel DNA sequencing, and we have more R loops at the beginning when NR4A1 is binding, and then after the replenishment of the serum, we have less R loops. And this is confirmed also using another type of sequencing using Prostig, which measures activity of nuclear transcriptional processing on the EEGs. We have EGR1, PROSB having high Prostig signals. So this is a summary of all of these projects. So under basal conditions, or NR4A1 is here blocking, acting as a traffic light on red, a red sign, blocking RNA-APOL2 and creating these R loops that pauses transcription of immediate early genes. Stress to the mix. This NR4A1 is released, and then we have a lot of aren't able to transcribe in these immediate-target genes. So when this stress signaling kind of give us, if IGs can be regulated by NR4A1 and it becomes adaptive proliferation, but if we do uncontrolled NR4A1 uncontrolled replication stress, there's DNA damage and mitotic defects, as you saw in the picture, therefore making the cancer less adaptable to our body, meaning smaller tumors. So this insight on NR4A1 has become a field of research in the lab after I left, because it has, you can imagine that they can have a lot of application of how to stop metastasis by controlling NR4A1. And with that, I'll acknowledge, of course, all the people involved in this project, mainly Hongsheng Guo, Dr. Guo, who did all the amazing work at the bench and gave me terabytes and terabytes of data to have fun and analyze all of these data that I showed, and more that I didn't show, because of course there's experiments that don't yield any conclusive evidence. And of course, the Daniel Haber and Shyamala Maharswara and the PIs of his lab, and Michael Lawrence, my postdoctoral mentor, which is the head of the computational lab in the MGH Cancer Center. And with that, I'll be happy to take questions from talk or for professional questions, advice, career, etc.
Great. Thank you so much, Dr. Golczer. That was very good. We have a few questions for you. So number one, how easy is it to transition from bioinformatics to biology or biotechnology or medicine back to bioinformatics and vice versa? Are there skills that are transferable?
Yes, I am a firm believer that we have to have a strong biology background. There are a lot of bioinformaticians that go in from computer science or even math that don't have that biology background. So having a biology background makes the transition easier. If not, if you have mainly computational skills, you'll have to make an extra effort to understand the biology behind what's going on in front of your computer monitor. So going back and forth, what some people already talked, how you're willing to learn all these tools, all these computational tools, or in the case of a computational person that wants to go into biotech and bioinformatics, how willing are you to understand how DNA replication works, how molecules work? That will give you a lot of insight and make the transition easier.
Great, thank you. What was your role in this project as a bioinformatician?
Well, I was a lead bioinformatician, so I got the data from the sequencing facility to the plots and the paper that you saw on the slides, and you can see. Of course, the project had already other bioinformaticians working tangentially, but when I started the fellowship, they gave me this, and they, especially, I remember this, Michael Lawrence told me, this is kind of a fishing expedition. We want to see what's in this massive amount of data. And the role that I play, as I say, is going back and forth with Dr. Guo to figure out what interesting questions could be asked from this experiment. So that's how I describe my role in it.
Great, thank you. I think we have time for one final question. So how useful was the experience you had working on this project for your current career?
Well, I think that so first of all the undergrad, master's, PhD gave me the basic skills to jump into this postdoctoral fellowship. But this project was basically what I think it landed me my job in industry, because I was working with insects, as you remember at the beginning of my talk. I was working with insects. So some people in the industry are like, oh, well, but you don't talk or study about humans. So they're a little bit hesitant. Once I made that jump to human. And DNA is DNA. Like it's the same for bases in the case of DNA, same for bases in the case of RNA, regardless of the system. But that jump and all these new technologies, I don't know if the people hearing or making the questions are familiar with ATAC-seq, PRO-seq, DRIP-seq. I wasn't familiar with those until I started with this project. So basically that gave me the skills to go into a more fast-paced environment in industry and work with the technologies that are just being developed like in a matter of months and be able to implement them into the insights that we need in the company.
Great. Well, thank you again. That was a very informative talk. I really appreciate you taking the time to come and speak with us today.
Thank you for having me, Laura.
Of course.